Good afternoon, everyone. My name is Jessica, and I'm going to show you part of my results on the pharmacological screening of venoms from 5 Brazilian microorganism species on different ion channels. Oops. As we know from the literature, the microorganism venoms are either predominant in phospholipase A2 or three-finger toxins. So if we take a look in this map here, we can see that the north to the further to the north of the American continent, we find more venoms predominant in phospholipase A2, and the further to the south of the American continent, we find more venoms predominant in three-finger toxins. That is the case in Brazil. So in Brazil, most venoms described to date are predominant in three-finger toxins, with a few exceptions. These two main toxin families, also known as alpha neurotoxins and beta neurotoxins, are main players to the envenomation outcome. And they are responsible for the neuromuscular paralysis, acting in the neuromuscular junction. They are damaging the axon terminal and impairing the cholinergic system. For example, inhibiting the nicotinic acetylcholine receptors. In this work, we study 5 different venoms from 5 different microorganism species, showed here on the left. And these specimens were collected in the south and southeast of Brazil. And we use the 2 electrode voltage clamp technique to test venoms and fractions in different targets and different ion channels. And briefly explaining the method, we extract and prepare the Xenopus laevis oocyte cells, and we inject with messenger RNAs and express the targets on the cell membrane. And then we measure using 2 microelectrodes, one to set the voltage and one to measure the current of the whole cell current. And then you obtain the results. The nicotinic acetylcholine receptor activities are a classic and important activity for the microorganism venoms, we decided to test the muscle and the neuronal types of the nicotinic acetylcholine receptor. So here, to understand the graphs, we have represented by triangles the pulses of acetylcholine, the agonist. And in the black line, we have the control, red line, the venom incubation, the blue line, we have the venom activity, and the gray line, we have the washout. So if we look at the blue line, we can see that all of the 5 microorganism venoms tested were capable to fully inhibit the muscle type of the nicotinic acetylcholine receptor. And they they show different reversibility. So for example, if we look at the Microornis corallinus venom, we can see that after the 3rd pulse of acetylcholine during the washout, this venom is almost totally reversible. And while for example, the Microornis decoratus venom, we can see that these venoms are not reversible even after 6 pulses of acetylcholine during the washout. And here on the right, we have the tests on the neuronal type alpha 7 of the nicotinic acetylcholine receptor. We tested 4 venoms and 2 of them showed activity on this receptor. And we can see here in blue that the venoms were capable to, the Altirosis and frontalis venoms, they were capable to partially inhibit these receptor and subsequently potentiating the receptor. It was more or less 50% of inhibition, and Microornis corallinus and Carvalhoi venoms did not show activity. To illustrate how potent the microorganism venoms can be on acting as inhibiting the muscle type of the nicotinic acetylcholine receptor, we tested a single fraction which contained just 2 toxins. As we can see here in the graph, this fraction, the single fraction alone, has enough potential to inhibit the muscle type of the nicotinic acetylcholine receptor. As you can see here in the first pulse of acetylcholine, that we see no activity on these receptor. Even not being conserved target, actually a reported target for the microorganism venoms, we decided to test the voltage-gated potassium channel 1.3 and 1.6. And here again, we have the black line for the control, the blue line for the venom activity, and the gray line for the washout. And we can see that all of the 5 microdose venoms were capable to partially inhibit the Kv 1.3. The microdose, and all of them were reversible after the washout. The microdose corallinus venom was the most effective on inhibiting this, the Kv 1.3, with more or less 50% of inhibition. And now here the test is on Kv 1.6, was a bit different from the Kv 1.3. We can see that all of the venoms could inhibit partially or fully inhibit the Kv 1.6. For example, Microdus altirrostris, corallinus e frontalis venoms were the most effective, with almost totally inhibition of the Kv 1.6. And we can see a different reversibility when we analyze each venom. For instance, the Microdus corallinus venom, we can see that after the washout, the inhibition was almost totally reversible. While the Microdus frontalis venom, it was almost totally, we can see almost a full inhibition of the receptor Kv 1.6, but this activity was not reversible after a long washout. These activities of the microdose venoms in voltage-gated potassium channels raise questions. That is, for example, is the inhibition activity on these voltage-gated potassium channels a conserved evolutionary trait of the microdose venoms? Is this activity an important feature for their envenomation strategy? Well, we don't know yet, but we are investigating further these activities for other venoms, and as well as in the nicotinic acetylcholine receptor. The activity on the voltage-gated potassium channels may represent and may be an asset for further pharmacological exploration. This work was done in collaboration with Dr. Professor Jan Tittegam from KU Leuven, and I would like to thank him as well as the other authors. I also would like to thank the Instituto Vital Brasil for providing the venoms, and my home university, UFRJ, and the financial supporters. And I thank you all for your attention. And I will be happy to answer questions.
Thank you very much. We have a question in the room? Yeah, there's a question.
Hi, Jessica.
Hello.
Hi there. This is Rita. Is there a particular reason why you chose those two potassium channel types to test, considering that you have a higher effect and a muscle type nicotine channel?
Well, the 1.3 is an important target for in the pharmacological perspective point of view, and the 1.6 a bit less, but we also choose because we were expressing this channel on the oocyte, so we decided to test to see whether these venoms could act in other types of the potassium channels, not just the 1.3.
Okay, thank you.
Welcome.
Jessica, it's Glenn King here. Just following up on that question. I think the point she's making is valid. These venoms are not going to get anywhere near the brain, right? They're not going to permeate the CNS. But so do you think it's working on potassium channels at the neuromuscular junction? And if so, my question is, wouldn't the two effects be counteracting one another? So if you block potassium channels, that would be excitatory because you prevent repolarization. And that would be counter to blocking a postsynaptic receptor, which would be depressive.
I'm sorry, I don't know if the internet is not working well, but I didn't hear very well your question. I'm sorry.
Sure.
Could you repeat it?
Yeah, I'll repeat it. So I'm just wondering whether if you're blocking a potassium channel, a presynaptic potassium channel, that would prevent repolarization, right? And that would presumably be excitatory. Wouldn't that counteract the effect of blocking a postsynaptic nicotinic receptor, which would be depressive? In other words, wouldn't those two effects counter one another?
Well, we don't know yet. We need to investigate further all the activities we found, but yeah, it may be well, in this specific experiment we measured on specific channels. So yeah, but in thinking about the whole venom, maybe this is a combinatory effect that we are seeing. I don't know if I answer your question. That it's still not a good to me. The I don't know if the problem with the internet. I'm sorry. Did I answer your question?
Yeah, no, that's fine. And I'll discuss it with Jan later anyway.
Okay. Sorry. Okay. All right.
Thank
Any
you.
other questions?
Maybe if someone wants to send in the chat, maybe it's better to answer. It's really not good to me, the audio.
Okay, and also if there's anyone, does anyone online have a question and want to unmute and ask a question? Or because of the connection, if you do have a question online, you may as well write it in the chat. So yeah, please do send your message to Jessica in the chats. You could also send her a direct chat online. So thank you so much, Jessica.
Okay, thank you.